Yes, good evening to all the participants. Welcome to the ninth day session on network pharmacology and pathway analysis in internship and certificate course in AI and ML approaches of drug and vaccine design. Before going to today's session contents, can we recap the last session topics? So, what are the exact contents we did we discuss on yesterday? You want me to give the hint? So, what did we discuss on yesterday? Yeah, come with your answers. That genome, genomics, proteomics. So, we have seen the types of genomics and we have seen the types of proteomics. Actually, I've taken you few tools. I mean, few databases and tools. Can you tell me a few tools of genomics, few tools and databases of genomics? Whatever we discussed on yesterday, or few databases of genomics. Yes, very good. So we have discussed about cake genome. Like cake genome, what else did we discuss other than cake genome? What are the different genome databases we have seen? Majorly, I have taken you to UCSC genome browser. You remember? Where is it present? UCSC. Support, isn't it? In NCBA, I told you, no? So how to explore to the NCBA genome. Directly, I've taken you to the NCBA and I've showed you the genome. I typed Homo sapiens and then I gone to 22 plus X plus Y. Okay. So the next question is, give me examples for the proteomics. So what are the databases I've shown you in proteomics? Very good. So in UniProt, we have seen proteome database. Other than that, do you remember? I've shown you something called Pride from EBI. Okay, the proteome database from UniProt is from UK. Okay, so these are the things. Other than that, we have seen so many PPI interaction databases, isn't it? We have seen PPI interaction databases which falls under proteomics. Okay, now let me go ahead with today's session because we have numerous content to see in today's session also. So today we'll be discussing on network pharmacology, system pharmacology, pathways, networks, interact on diseasome, network visualizer, and some case studies. Network pharmacology. What is network pharmacology? It is actually a part of a network medicine. That is for building, for developing the network medicine, we are using this network pharmacology. And if you see the definition of network medicine, it is the application of network science towards identifying, preventing, and treating diseases. Okay, so for this network medicine, we have to take a set of gene interactions. Okay, whatever genes present in the network, instead of simply relying on a single gene, we also focus on the interacting genes of the particular hub gene, whatever major gene is responsible for the disease. Okay, instead of simply taking the major gene responsible for the disease, we also find the interaction patterns. If you see the interaction patterns, we can see one gene is positively upregulating, the other is downregulating. So both the upregulating and downregulating plays equal role in the disease part. Then we have to take based on the expressions, which is having more expression. So one should understand that instead of simply relying on a single target, we go with that single target interacting targets. Okay, if it is a PPI interactions, and similarly, you can see for the disease networks. one disease can lead to the comorbidity of another disease. Then what genes present in the one particular disease can lead to the subsequent disease. So everything, these are also a part of a network medicine. Of course, this comes under network pharmacology. Okay, so here majorly you can see we study in the form of protein interaction networks, metabolic pathways, etc. in this network medicine. So everything interactomics, all the kind of omics also comes under network medicine. If you see one such a kind of a drug for Zolgensma, this is manufactured only by Novartis. Only Novartis can able to come up with a kind of a medicine to treat spinal muscular atrophy. So the price of this single shot medicine, okay, the mentioned as 5.5 ml, is 18 crore in 2018, I believe. Now it is more than 24 or 25 crores. So one shot comes around this much price, and only one company can able to produce medicine for this disease, spinal muscular atrophy. Okay, and the major target, if you see, human SMN protein. Okay, so do you understand how much important is to work with these kind of a drug and vaccine designs for treating a single this kind of a disease? This much for us. So actually, this works under gene targeted delivery. Gene targeted delivery. If only if you know what is the exact gene responsible for the disease, okay, that code for SMN protein, and what are the interacting genes responsible. So this is most important to bring such a kind of a medicine for the disease. Now about network pharmacology. If you see the major, in network medicine, you can see a part of is a network pharmacology. Here in network pharmacology, instead of simply discussing on genes and proteins in the network, we also focus on the compounds. The compounds are also one of the fundamental inputs. We use it in network pharmacology. Compounds, for example, if you know, I am working on a plant called Boerevia diffusa. Boerevia diffusa is a herb which consists of more boerewinone compounds. Boerewinones are a form of retinoids. Retinoids, it means that these are isoflavone compounds. These compounds are responsible for treatment of kidney stones and urinary infection, because this plant also called as Kumarnava. So if you see these boerewinones, you can able to see around 16 boerewinones, I believe. So out of 16 nodes, if you want to screen which is the best one, okay, we can able to screen the best target the compound, as well as one target the interacting compounds. So we have to first screen the best compound for the important target, and then after screening the compound, we can go with the interacting targets, and we can see the binding patterns through docking studies, as well as stability patterns through simulation. Then we can conclude it. In this example, they are majorly taken for treatment of obesity. If you want, how to come up with a drug for treatment of obesity. So for obesity, we have different plant and we have set of compounds. And from the compounds, we take the best compound which is screened, and then we target the particular receptor for obesity. Okay, and then we come up with this. Okay, and here if you see, other than this, in this network pharmacology, whatever the targets we have taken, whatever the screen compound we have taken, we will be checking the PPI networks in string database. Only in the string database we can come to the top up regulating and top down regulating genes. Srinidhi Varadarajan, you can add the participants. I told multiple times participants to accept my invitation. They're not following. Okay, PPI network. If you see this PPI network, okay, if, we can see the databases like String DB, Stitch DB. These are some of the databases. Are you great? So whatever databases we take, we have even for few target proteins, we have the interacting proteins is more than 1,000 proteins. So from the 1,000 proteins, we can take top 100 upregulating, top 100 downregulating, and then we can screen based on the pathways and whatever is most important for a particular stage of a disease. That also we can find. For example, if you see this cancer, we have 0 to 4 stages of cancer. So the third and fourth can't be cured because of aggressiveness in abnormal cell proliferation. If so, we can able to cure from 0 to second stage. So what is the gene uh, present? What are the important genes present in the second stage? What are the important genes present in first stage? Like this also we can able to identify, and even we can able to check which are the most important genes based on upregulating and downregulating of expression. Okay, with the major target protein. Target protein, we also call it as a hub protein. Like this, you can able to see, we can use the gene enrichment analysis for enriching the genes, that is filtering the genes based on pathways, based on the organs, based on types of disease, etc. And then we finally, whatever we have taken, we can validate the results through in vitro as well as in vivo. In vitro, we can check with the particular cell lines of what disease we are working, and in vivo, we can check with mice, rat, etc. Generally, for obesity, the best animal model is to check is the rat. Okay, so like this, we can try with for a particular disease. In this case, it is obesity. So like this, we can try with any kind of diseases. Okay, we can take curcumin from curcuminoids, curcuma longa, and then we can try to make it. We can also, instead of taking a single plant, we can also take multiple plants, and from 2 or 3 plants, we can take active compounds. So earlier I mentioned to take only single plant, but here even in the network pharmacology can extend to work with multiple herbs. That's what happening in Ayurveda. Ayush. Did you heard about the Ayush? Okay, so what is Ayush? Can someone expand the terms Ayush? Or you can unmute and answer. What is Ayush? Because we are living in India, we should know about these terms. And our course is related to, and the eminent scientist is also here from Ayush. We can also learn a lot from her. Yeah, what is Ayush? Expand Ayush. One should not forget about impact. Im, I believe impact is this. I check the spelling. IMPPAT. So one should not forget about this impact database. Okay, and the Ayush website. Now, lot of funding opportunities are there. Okay, the Ayush also the department merged with the Bureau of Indian Standards. Bureau of Indian Standards has their own kind of a department called Ayush. They're also working on developing standards for the herbal formulations in the herbs, etc. Very good. The expansion is Ayurveda, Yoga, Naturopathy, Unani, Siddha, and Homeopathy. Excellent, excellent. Even I found new participants are interacting. Very good. So one should remember that whatever we can take in this Ayurveda for treatment of a particular disease can be uh, treatment of SARS-CoV-2 infection. We use Kabasira Kudinir. Kabasura Kudinir. Did you hear about this? No, it's a combination of around 20 herbs, 16 to 20 herbs you can find. So whatever we find in that herb, we have multiple compounds, nearly minimum 20 compounds in a single herb. Then how about if you take 20 herbs, 20 to 2400 compounds will be there. Okay. So whatever compounds we take, even if you want to check which is the best compound which can works on the binding of spike glycoprotein, in SARS-CoV-2 infection, the spike glycoprotein is a structural protein, is one of the structural protein. There are actually 4 structural proteins in SARS-CoV-2 virus. And similarly, we have 16 non-structural proteins. The proteases, polymerases, everything comes under non-structural proteins. So totally 4 plus 16. So totally we can able to find around 20 proteins are available. Out of 20 proteins, spike glycoprotein is one of the important target. That's it. But few compounds, whatever I take, like if one compound is majorly targeting the open conformer, the other is targeting the closed conformer of spike glycoprotein. There might be the few compounds can target the polymerases and proteases. Anyhow, we want the inhibition of SARS-CoV-2 in transcription or translation stage. Even the mechanism, the drug mechanism of these compounds can also vary. And just because of that, we can tell it as an effective remedy for preventing these kind of infection, isn't it? So we can also take, so whatever we work with oils, we can implement the network pharmacology, and this is some of contemporary research happening in the bioinformatics world, that is in the development of drug and vaccine research. System pharmacology. So till now we discussed about network pharmacology, network medicine. Now we are discussing on system pharmacology. System pharmacology is the application of system biology. Like how we find the network biology is the application of network medicine. So similarly here you can see like this. And what is pharmacology actually? The pharmacology, like mechanism, if you still want to study the mechanism of action, adverse reactions, side effects, okay, all these things comes under pharmacology kind of a study. System biology. What is system biology? So if you want to understand system biology, uh, better remember the types of omics studies. Study of the omics, genomics, proteomics, interactomics, and other than that, pathways, networks. So these are all comes under system biology. Okay, so there is an association between this system pharmacology and system biology. Now, let me go ahead. So it seeks to understand how drugs affect the human body as a single complex biological system. Okay, and if you see this system biology, I told you it is a combination of study of all the types of omics, and of course, we need the molecular structures, especially the targets, to take a gene, the gene code for which protein, and after that, what is the structure of the protein, what are the different conformers of the protein, okay, what is the size, and what are the binding receptor binding domains of the protein. So like this, we need entire details of the structure of the protein. Then what we can able to understand even whatever drugs we have designed is going to target in the appropriate binding sites or not. A drug should not bind in any other sites. It should target only the binding sites where it can give the biological effect. So, studying about all this kind of omics along with the structures and also gene networks, okay, everything comes under system biology. So here in this right side, you can see a picture of a metabolic pathway network of E. coli taken from cake website, that is genome.jp/cake. In system biology, we can also see a part of a study called biomolecular computing. Generally, in system biology, we study cellular computing. The cellular computing is the generic term. IT department, EC department, everyone can use this generic term called cellular computing. So under cellular computing for the EC, that might be a cell, a battery cell. But for us, the cell is can be our mammalian cell, or it can be a bacterial cell. Or if you are studying on biomolecules, then whatever cell we call it as a biomolecular computing. In biomolecular, we can also study any particular species. If you are studying on bacterial biomolecular, then it is called bacterial biomolecular computing. So what is this exactly? So one should remember that as a computer. If you remember a simple computer, so we have a CPU, and the CPU is linked with a keyboard, mouse, etc., which can give us inputs, and the output will be delivered through the monitor, projector, printer, scanner, like this, isn't it? Similarly, here if you see the bacterial cell, okay, what exactly the input can be taken by the bacterial cell and what kind of outputs it can able to release. So this we can also link with neural networks. We can also correlate the inputs, weights, etc., and use a correlation to calculate the corresponding output. What is the amount of fluorescence a bacteria can able to exhibit based on the uptake of light, based on the uptake of light as well as uptake of lactose. So some bacteria also exhibit fluorescence, especially Pseudomonas fluorescens. The name itself can able to see Pseudomonas fluorescens. Okay, can exhibit their fluorescence based on the light and based on the thing medium, how much it has taken. So based on that only it can able to show that. Okay, like this we can see quorum sensing and producing the electric current. The even bacterial cells can produce electric current. So that depends on the temperature pH in which we are growing this kind of cells. So other than as I mentioned, we can study these kind of a computing in the form of molecules, can be a gene or protein. We can also use this instead of bacterial cell, we can use organelle. We can also use organism. So everything we can be able to study. So whatever we study is a part of a cellular computing, and of course cellular computing is a part of system biology integrated model. It's a part of a system biology. Okay, so like the system biology is like a version where you can study numerous things, like uh, producing a PDB file, I mean, generating a PDB file for a protein structure from the X-ray coordinates is a simulation. And if you want to check the stability of ligand and receptor binding, that is also a simulation. If you want to check the effect of solvent of in a particular protein, that is effect of multiple solvents, each and every solvent to a protein, that is also simulation. So whatever we are working, like if a person is consuming alcohol, what is going to happen with that protein, that also we can simulate in the computer. Got it? So whatever we can try in our body, if we can able to work in silico, it's a simulation technique. Of course we study with respect to period of time. Over a period of time, what is going to happen? Okay. Okay, now let me go ahead with this topic called network biology. Network biology is a combination of biology, network theory, and statistics. Okay, of course, network biology is also a part of system biology. Main parts of network biology. So before that, I just give you an outline. So that is in the 1999, from 1999 to the 2005. So you can see many important genomes got published. The human genome project drafted in 2001 got published in 2003, and even the rat and mice genomes are also published in the subsequent years 2002 2003. Okay, so most of the important genomes related to the treatment of human diseases I can able to find. The genomic era, if you ask me, I can tell 99 to 2005. And from 2005 to 2010, many of the protein interaction networks came to existence. It got rapidly expanded after even 2010. And then you can see after 2010, many scientists worked on the interactomes, RNA. Even the RNA, non-coding RNA, can also play an important role in the development or progression of disease. Then after 2010, many of them are also worked on interactomes. And in 2015 onwards, many are working in the system biology approach. So this is one of the contemporary approach for coming up with a precision medicine. So networks, if you see the networks, we have types of networks like PPI, protein DNA network, gene correlation, gene regulatory, metabolic network, signaling network. All are different types of networks. Gene correlation is different, gene regulatory is different. Okay, like this, I just showed you the major thing. We also have the carbohydrate protein interaction networks, lipid protein interaction networks. Like this also we have types of networks. Okay, but whatever I shown are the major type of networks, and this is a metabolic network, and this is a protein interaction network of yeast. Can someone tell me how much percentage of coding genes, how much percentage of coding genes or codons are present in yeast? How much percentage of coding genes or codons are present in yeast proteome, I mean yeast genome. Percentage of coding genes in yeast. The next is human. Because we just completed genome, one should know about these kind of information. Of course, this is a national competitive exam question. Even how many genes are present in human, what is the size of human genome, this is also you can find in GAT question in the previous years. 1 to 2 percentage is the coding gene for human, that is right, exact answer. Not more than 2 percentage genes only we can see the code for proteins in human. It means that 90, more than 98% of the genes or non-coding genes are also called as introns. That was lazy. Our genes are not coding for the proteins. 98% are not coding for proteins. How about for yeast? How many percentage of coding genes are present? More than 99 percentage. More than 99%. See, that's why you can see this a single unicellular organism is more effective to produce many proteins. So if you want to produce even many of the recombinant proteins, we try with this kind of yeast next to the E. coli. Okay, next Saccharomyces cerevisiae can be used. Okay, now let me go ahead. Gene regulatory network. So, what is the difference between the gene interaction network and gene regulatory network? In gene regulatory network, we can see two different colors. So, one is the red color, the other is the black color. The red color is the positive regulating gene. The black one is the negatively regulating gene. Okay, so in the center you can see a gene called TP53, one of the most important target for many types of cancer. So this TP53 is linked with so many genes, but few whatever shown in the red are positively regulating and whatever are shown in the black or negatively regulating. And now how this networks are created. So related to this, a small theory is mentioned here. If you see in this picture, okay, there are actually three nodes. Whatever you can see is the node. First node, second node, and third node. If it is gene-gene interaction network, the nodes are genes. If protein-protein interaction networks, the nodes are nothing but proteins. So if this is node 1, you can also see the edges. The edge, the whatever you see in the form of lines or edges. Okay, the edge can be connected to the gene itself. The edges can also be connecting the different genes. The first node is connecting to the uh, second node through this edge. This is the edge for 1 to 2. But if you see the 1 to 3, we have three different paths. The shortest path connecting first node and third node is this. This is number 1. This is the base path because it follows the shortest thing. The best should follow the shortest path. And the next to that, what you can tell, this one or this one. If you tell this, this is correct. Okay, this is the second best one because of shortest path. But this third one is going to have a lengthy line, the lengthy edge. So this is the third. But if you want to go to one place to the another place, you may choose the paths based on the traffic or based on kind of hurdles, whether no diversion, like however you see in the Google Maps. Sometimes you won't prefer to follow the shortest path. Sometimes you prefer to follow even the longest path based on the traffic, etc. Isn't it? Similarly, even our genes function similarly. Okay, so based on the kind of metabolic flux, okay, so the genes can take various edges, that is various paths in the pathway. Got it? So sometimes like based on the disease progression, instead of positively regulating, it can negatively regulate. That can also happen. I told you the red is positively regulating and the black is the negatively regulating. So the color can also change. Okay, this is something about the simple graph. And one more question to you. If I want to develop a network graph, how many minimum nodes we should have? If I want to develop a graph, if I want to develop a graph in the network, how many minimum nodes are required if I want to draw a graph? How many nodes? Yes, very good. The answer is three. Okay. Why the answer is three? At least three is needed, isn't it? If you have just two nodes, if we have two nodes, whatever I connect the center to the another center of the node, this is just a line. We can't call it as a graph. Unless you have another node, you can able to find the form of a kind of a cyclic process, a cyclical kind of pathway. This is a cyclical pathway, so we should need minimum of three nodes to connect a graph. Okay, and now let me take you to the other information. So in system biology, we try to connect different types of omics. So if we know the genome, we can also get a few knowledge on proteome, like whatever genes, coding genes which code for proteins, that proteins can will be interacting the protein network. So we can have a kind of a relation between the genome proteome, and also we have hyperlinks in the genome to connect with the proteome, and similarly metabolome, etc. If you see uniprot, the uniprot has the entire thing. Okay, it can link to any of this kind of an information. And yeast protein networks, you see, whatever a group of proteins in this network, this can follow one pathway, and whatever you find in this can follow a second pathway. Okay, for example, if you see metabolysis pathway like glycolysis pathway, gluconeogenesis pathway, citric acid cycle pathway can be somewhere nearest to that. You got it. So like this, you can see in the metabolic networks. However, the metabolic networks, you can see metabolites are linked in different pathways. Even for the protein expressions, in the protein, you can say something like NF kappa B or some other kind of NF kappa B, WNT. Like this, you can see different proteins can also have different pathways and can be interlinked with each other. Gene regulation network of CRG. Okay, so however we find the black lines, the red lines, green lines, that can give you the kind of expression patterns. Metabolic flux, how the metabolites are linked with each other. Okay, and even you can find multiple pathways we can able to observe. Oxidative pentose phosphate pathway. So, and this comes under gluconeogenesis and pa- and then oxidative pathway, oxidative pentose phosphate, and in the bottom we can see non-oxidative pentose phosphate. So whatever you can see is a different pathway here, and Embden Mayer pathway. So if you see, these pathways can be interlinked with each other. Like however you see in different proteins can also have a different pathway, and even the pathways can have a kind of relation. Similarly, here the metabolites can also have a relation and can be interlinked with each other. This is a metabolic pathway of equilibrium. And however, I mentioned, if you see in the keg, the glycolysis can be linked with the ED pathway, pyrrose phosphate pathway, the Krebs cycle, the TCA cycle. So, like this, you can see all these pathways can also be interconnected. So, you have numerous metabolites interlinked in this pathway. So, pathway role in target identification. I told you, instead of simply restricting up to a single target, just go with the connecting targets, and even you take that as one of the important target for your development of drugs. If you want to develop a drugs, instead of simply relying on single protein target, go with the network of proteins as that also significant targets. And for network databases, we can go ahead with StringDB, Stitch, Funcook, Impact, Hub 2.0. I show you, like we can also, if you go with the string database, we can able to go with the gene enrichments through any of these biological process, molecular function, cellular component, cake pathways. So, for example, yeah, if I search for a particular protein, okay, especially I work on a particular cancer called hepatocellular carcinoma. For the past 5 years, I'm working in this cancer. And if you see, whatever I work, okay, so the most important organ is liver, which I can find from the tissue expression. So, tissue expression is one of the gene enrichment information. Like this, you can find whatever is more important. You can take a pathway and you can take a pathway related to the hepatocellular carcinoma. So like this, I can able to do the kind of enrichment. When I selected the liver, I can able to see for my target what are the important proteins connected to the liver. And now I have taken two different filters. Other than liver, I have taken one more gene ontology and selected hepatocellular carcinoma. Liver is shown in violet color and hepatocellular carcinoma in this red color. So whatever you find the blue and red are the significant targets for our protein when compared to the red alone or blue alone. Okay, so like this I can able to find the information, and from this I can able to see these are five important targets, so which we filtered from the protein interaction networks. And if you see the stitch database, so other than finding the protein interactions, you can also see the drugs interacting with proteins information also available. Celoxocab, indomethacin, how these drugs are interacting with the proteins. This information, and I work with this kind of a plant compound. Even the interacting proteins information also available in Stitch DB. That is compounds or drugs interacting with proteins. And here also I can check for a liver. What is the important target for the liver? Like this also we can check, and that alone is shown in red color. And this kind of an output I am showing in the tool called Cytoscape. Cytoscape, this is a freeware. This can be used for visualizing any type of networks, protein, gene, or carbohydrate protein, whatever you want to see, all the kind of metabolite network or whatever networks I've shown, everything we can able to visualize in this Cytoscape visualizer. This is a freeware. So if you show the earlier things, you get in trouble with plagiarism. But whatever you can open in the Cytoscape and you go with the gene enrichment, so all these things can don't lead to plagiarism, and so can use this output for publications. In Cytoscape we have so many apps. We have multiple apps. We can install the apps in the tool itself, the software, this open source software. Whatever you want to work, we have many apps. Apps are nothing but programs. Okay, so I have installed one app called CytoHubba. Okay, so I just click that and I search so many apps, and if I want to work with the CytoHubba, I install so I can able to find it here. So then whatever I select the CytoHubba, I can work with whatever I want. Similarly, you can try the significance of other apps. So to know more about the apps, etc., you can use the help option. Okay, so video tutorials are available as well as normal kind of API documentation available. So how to use the Cytoscape. Now about databases. Where exactly we can find this network information in the databases? I told you the most important database what we have to look through. This is an indigenous database developed from our nation. The Indian scientist has brought this kind of a database to check not only about the compounds present in the Indian medicinal plants, but also what are the genes like the compounds interacting genes information is available, and even diseasome networks, like the plant is used for treatment of what diseases. So entire disease information, like whatever is can able to see all this information you can able to see from this impact database. So for example, so I searched for I believe a plant. So whatever plant I searched, okay, plant, I can able to see that plant can able to treat this much diseases. Not the entire plant it can be used for these diseases. It can isolate a specific compounds. Might be a phenolic compounds for treatment of one particular group of diseases, and the tannins can use for treatment of another diseases. Okay, so whatever compounds present can be used for treatment of this much number of diseases: eye infections, anuria, asthenia, anticonvulsions, and anti-inflammatory, hypoglycemic agents. Like this, we have find different disease information. That is an you not only can see the disease interconnecting thing, we can also see the gene interactions also, the interaction of compound with the genes. That also is available in impact database. Hub 2.0, you can see I just search for a plant compound called Vitafrin A. I can able to see how this plant compound is treating the is is treating the, I mean, is interacting with the different proteins. And if you see this hub 2.0, you can also able to see the most important targeting genes for the particular compound. Network analyzed. So network analyzed is another tool. This is used for comprehensive gene expression profiling via network visual analytics. Okay. So for network analysis, we have to go through this URL, and here you can able to see the gene expression table. So in Express Analyzed, we can also link with a programming called R program. So through the R programming, we can able to analyze through statistical graphs, etc. Okay, like whatever gene expression data we have in the form of data sets that can be analyzed using R program. Okay, so how the gene expression we get the gene expression data? What are the wet lab techniques we can use to generate the gene expression data? How these data are posted? Gene expression, earlier we try with microarrays. Nowadays, many of them are trying with RT-PCR. Okay, so but whatever data sets we find, so most of the data are from the microarray experiments. So, like for a particular cancer, ovarian cancer, how many genes are responsible for a particular stage? Like this, we can download the data sets and we can use the R programming like this. We can import the libraries, okay, and then we can use the functions, and then we can able to find these kind of a graphs for that. Okay, we find plenty of graphs generated from the data values present in the data sets. Next is the metaboanalyzed. Metaboanalyzed, we have different metabolites and how these metabolites are linked with each other. And however we check with the genes, we can try with the same with the metabolites. Even the metabolites are also important related to diseases. For example, these are a raw spectra to the biomarkers, patterns, functions, and system biology. So this information you can able to find. Even the metabolites can increase the progression of a disease. So one should know what metabolites are produced. And of course, these are linked with the enrichment analysis, pathway analysis, network analysis. However, if we find the for the gene, even for metabolites, we have a similar website for this. The next what we discuss is the DigiNet. DigiNet is for disease gene network databases. So if you go to this DigiNet, before that I'm just showing you the type of tools for visualizing the networks. Cytoscape, gadget, and graph-related programming, you can go with LADA, NAUTI. Types of networks, random network, scale-free network. You can have any kind of these kind of networks in the databases. There is also a kind of network called Interactome. So after completing this, I go with the Digi network. I have few slides also with Digi network. I explain about that. I'm showing you a few more things what I missed. Interactome. The name itself implies in protein-protein interaction. If you find, I mean, if you want to check the proteome, we work with protein-protein interaction networks. But in Interactome, we find the RNA networks, gene regulatory networks. Okay, so these are the major things you can find in Interactome. Now, diseasome, I told you human disease networks, also called as diseasome, you can find in this disease network. Earlier in IMPACT, how a plant compound can be used, I mean, can be used for treatment of various diseases. That alone we have seen. But entire details related to the genes responsible for the particular diseases. Up to this, if you work, this is what we call it as diseasome. Okay, so to check this information, you can see few more information about the diseasome. So we have something called shared gene formalism. That is, one gene is linked to two different disease phenotypes. For example, if you see TP53, this is most important gene for many cancers. One target is linked with two different diseases, two different cancers. And we have a second category called shared metabolic pathway formalism. says that if a metabolic pathway is linked to two different diseases, then the two diseases likely to have a shared metabolic origin. Example are metabolic disorders. Okay, we can also see disease comorbidity. Disease comorbidity, like for example, if you see cervical cancer, how the cervical cancer is caused because of viral infections. What kind of viral infections? If a person is human is suffering from 99% of HPV, human papilloma viral infections, obviously they are more prone to receive this cervical cancer. Some viruses are linked with the cancers, a kind of a comorbidity. This is what we call it as comorbidity. One disease linked with the another disease. Okay, like handicap patients more subject to get blood pressure because of stressing. They have more blood pressure, isn't it? Handicap patients, those who are suffering from polio, if you see like this, okay, not only we can see with respect to viral, with respect to cancer, comorbidity, we can link with this kind of autoimmune diseases with respect to analgesic, I mean, the pain related diseases. Now, let me go ahead with the TG network. Given in this TG network, we have genes, different genes, what are responsible for a particular disease and how one gene is linked with another disease. All that's what I mentioned, isn't it? That's what you can find in the form of networks. So this information you can download in the form of an Excel file data, and this you can also analyze using the R package. Also, this data we can open in Cytoscape. If you open in Cytoscape, you find the form of network. In Excel, if you open, you find the form of data. In R package, you can also get in the form of graphs. And we can also link with the Python programming, Perl programming. So whatever data we take from the GNet is can be worked with these kind of languages. So there are some specific terminologies in the disease zone. For example, disease specificity index. What is disease specificity index? There are genes that are associated with multiple diseases. Example, tumor necrosis factor. Tumor necrosis factor is, of course, an important protein. If you have more tumor necrosis factor alpha, we are not prone to cancer, because the name itself implies this is the most important protein, a group of proteins responsible for combating cancer. Like how the immune cells can combat cancer, even these proteins alone can also combat the cancer. But the same tumor necrosis factor, if present in excess amounts, can lead to autoimmune diseases. Okay, we should not have more TNF alpha. We should not also have less TNF alpha. So this is the nature. The regulation is there everywhere. Okay, so if it is linked with the multiple diseases, okay, then obviously we can able to calculate that particular target gene information in the form of disease specificity index. Say a formula is mentioned based on what diseases that particular TNF is linked. One is positive, one other is negative regulation. I told you an example. Like if you can see, there is one more index. We have so many indexes like this to check in disease zone. We find these kind of a data, but one should know what is disease specificity, what is disease pleiotropy. The name itself implies pleiotropy. What do you mean by that? One gene is responsible for multiple diseases. Like however I told, TP53 linked with ovarian cancer, cervical cancer, or liver cancer. Okay, that is disease pleiotropy index, and this is like this. I have a terminology. So if you go to the disease chain network and if you search a particular SARS-CoV-2 as an input, if you search, you'll be getting so many genes responsible for the SARS-CoV-2, and for that you can also find the kind of disease specificity index and other pleiotropy index, etc. Okay, even you can search for a spike glycoprotein also instead of searching a particular organism in that diseasome network. Cytoscape, I told you, this is an open source bioinformatics software to visualize any kind of networks. So here we can able to visualize any kind of networks. More about the Cytoscape, You can search in Google, you get much information. So if you want to go to the Cytoscape, go to the cytoscape.org. So Cytoscape only one software, only one exe file. After downloading and installing, you can install the apps inside the Cytoscape software itself. These are the icons of the Cytoscape. Each and everything is a kind of a program to work with the Cytoscape. Okay, that's what I call it as an app here. For data visualization, you can see. So the East change in networks, you can see like this. Okay, in the Cytoscape, this is actually a demo file I just opened and showing it to you. And this is related to colorectal cancer, the different TCGA database. What are the important genes responsible for colorectal cancer from a particular database? And there is something called gene enrichment analysis server, GPIA. So this is here you can able to find the differential gene expression for any particular target. For example, HSP90 alpha. If I give that as an input and if I search what are the interacting genes of HSP90 alpha, and how about the differential gene expressions, whatever gene it is going to interact, how about the positive and negative expressions that you can find in this GPA server. We can also go with gene enrichment through enricher server. If you type a single target and if you want to increase how many genes you want to search, top 100 or top 200, we can fix a particular value and you can find all the genes, whatever is most important interacting genes in the network. So this can also be used as a gene enrichment, enricher. Case study. So I believe I've already circulated this paper, ovarian cancer. In this ovarian cancer article, if you see what exactly I've tried is, I have taken the most important genes responsible for ovarian cancer based on multiple immunological signaling pathways, and then I screened one particular herbal target from three compounds. So initially, I worked with identifying top 100 differential expression genes through fold changes and the p-values. You can see the fold change and p-values. So top 50 upregulating, top 50 downregulating. So totally 100 differential expression genes. Then I have gone for gene enrichment through different pathways. There are pathways like PA3, AKT, RAS. So how about the the p values for this? So that I have mentioned, and what is the most important, we can able to see from the intensity of the colors. And the gene and that red color and green color shows the red is the positively expression gene and the green is the negatively expression gene. If I take what are the positively and negatively up regulating and down regulating, we can see in the form of colors. So like this, we can see what are the top ranked genes, what are the top ranked genes and the enriched pathways. You know, for the ovarian cancer, this SART1 is almost interacting with 18 different proteins. So only we find 19 as the number. So like this, you can see almost 100 genes we can able to check how many genes are interacting with each other. And if you see this overall top ranked enriched pathways, that also we can able to find, and for that p values can be determined. And even we can go with immune system related enrichment pathways. So okay, so from the databases and the tools, we can generate these kind of a data. Other than just going with the top 100 regulated genes, that is top 50 and upregulated, top 50 downregulated, I also increase the size to top 200 genes. For the top 200, how about the top 100 upregulated? And what are the top downregulated? This also is analyzed. And I have used again different enrichment filters. Okay. And if you see here, after this, whatever I identified as the top 10 genes that I've docked with the three plant compounds. I earlier mentioned you in the molecular docking. The quercetin is docking 6 out of 10 targets. So I have concluded that quercetin is the best target for treatment of ovarian cancer. The next is case studies too. Here also I've used the similar kind network, the system biology approach. So earlier I tried discovering a drug for treatment of ovarian cancer. But here in this kidney cancer, I've used the data sets. I've downloaded the gene expression data sets, and I just focused on to treat, to diagnose what are the important genes in the stage 1 and stage 2 of kidney cancer, that is renal cell carcinoma. Because if you go with the third, fourth, no use, because already you find it is badly damaged. The kidney has to be transplanted if it is advancing to the third stage cancer. So up to 1 and 2, we can able, if we can able to diagnose the gene, we can able to treat. So I just limited to that stages of cancer and found the important genes response. Okay, so this paper also I've just I think I uploaded in this WhatsApp. Okay, so I have completed today's session, so I can go with some kind of illustration of the databases. Impact database. Did I illustrate you like how to use the impact, meta-analyst, etc.? Yes or no? I just want yes or no. Can you admit an answer? Because what to show, what not to show in the limited time, you have to decide, not myself. You want me to show yes or no, or type in the chat box. If no, I can go with some other kind of illustration of some other thing. Yes, you want me to show. Only one is mentioning. How about the others? Can I show this illustration or I can go with MATLAB tool? I also have different plans if no. Yes or no? One more participant? No. Okay. She want the MATLAB when I asked about the MATLAB. Please show the illustration from my side. Okay. So the first come, first serve. You're right, sir. Now let me go with the impact, but net is too slow. Okay. Metamo analyzed, and we can also go with the network analyzed. Network analyzed .ca. So click here to start. So here we can go with Homo sapiens. We can specify some gene, or we can try with the examples. In this gene list, we have endotoxin tolerance gene signature. 99 genes are available. We should not paste a single gene. We should paste some combination of genes. Already some examples are available. If you don't know, go to the enricher. You find the HSP, some kind of a protein, whatever you want to search, you search about protein. You get top 100 proteins. Take that one protein along with the 100 proteins, copy and paste it here. Okay, now I just pasting the sample already present here. Okay, so whatever I have uploaded, so we can try with it. Okay, upload. You have to click upload. 99 chips has been successfully mapped to Windows. You can click to proceed. You can click then proceed. That's it, proceed. Yes, proceed. So now we are getting some information. I just increase the size. So protein interactions, tissue specific, tissue, any specific tissue. If it is related to adipose, subcutaneous. So please then click proceed. Proceed. You can also select a few more things, whatever you want. We want to check for the protein drug interactions. We select and we can proceed. The protein and drug target was collected from the drug bank. Okay, no problem. It is asking everything before proceeding. So in network analysis, what exactly can find? Gene expressions. This gene expression, how we want to analyze. Okay, we have a tool here, but still, if you want to download and you want to work with the graphs, we can work with R programming also. So you can able to see this topology. Degree distribution takes some time to check it. So if you want to download particular SIF, you can download and open the network. But this is a zip file. Okay, I go with the small photo. So this is the information we are getting from this. Okay. Now, if these are some information you are getting from the network analyzed, now let me take you to the string DB. I think I didn't show you much information on the string DB. Now I'm going to show you. If I'm working on a HSP90 alpha protein, I type HSP90AA1 as HSP90 alpha, and I'm going to search for the Homo sapiens of HSP90 alpha. And if I want to get a top 50 interacting proteins, so HSP90 alpha is present along with these 10 interacting proteins are there. But if I were focusing on a particular disease and I want to increase the interactions, first I have to go to the settings. In the settings, instead of 10, I can give it the 50. And if you want to increase it to 100, you can increase it to 100. So whatever I given as 50, if you click update, so 50 interactors, interacting proteins you can see. And then if you want to go with the gene enrichment, okay, so or for a particular disease, for example, myself, if I'm working on prostate cancer, I click the prostate cancer. Okay, whatever is selected as prostate cancer is in this orange color. The next two, if I want to use the heat shock protein binding, this is selected in violet color. So orange and violet colors, whatever you can see, this is what exactly we have enriched according to that type of disease as well as the binding patterns, particular binding patterns. Okay, this is how whatever two things we find are the most important things. Two different colors, only one we can see. Okay, so like this we can able to go with this kind of string DB database and the Indian medicinal database impact. Just I take 1 minute to complete this. Herb 2.0 can you can also try with that. That is a Chinese database. There also you can find this kind of information. However, I try with the impact. For example, if you are focusing on phytochemicals, and you know our company is majorly involved in this kind of development of secondary metabolites. If you want to work on, I mean, a particular metabolites like quercetin, curcumin, methafrine, ethanoid A. So like this, I am marketing around 26 different secondary metabolites. So in our lab itself, we are synthesizing. And if you want to get even nanoparticles, we conjugate with the nanoparticles and we are providing these products. Whatever we provide is above 95 to 98% purity. And if you want with less prices, we can also give 50%, above 50% pure compounds, above 80% pure compounds. Whatever compounds we provide, this can be used as a research chemical grade chemicals. That is, we can try with in vitro studies and in vivo studies. Humans can't consume these kind of drugs, but we can try with cell lines. We can try with the mice, rats, etc. The approval is available. We can also provide certificate of analysis. For example, if you search for a Tulasi plant in Indian medicinal plant in IMPACT database, okay, we can able to, if you know a particular compound, you can type the compound, or just give a medicinal plant and you search, you get a numerous kind of compounds present in the Tulasi. So omifoliol, sparsiflorin, so these are the compounds present. If you want to go to a specific compound, you can go to the phytochemical compound to the identifier or name, and you can get more information related to drug likeness, ADMET. So for a particular compound of a Tulasi, okay, a kind of a phenolic compound, we can able to see the Lipinski rule, 5 violations, no violations, and even it is satisfying the other kind of a drug likeness filters. Okay, and even the can able to see the drug likeness score as 0.7, and you can also see the admit properties. Okay, like however I search with the Swiss ADME, similarly for each and every phytochemical, the physicochemical properties related to drug likeness, pharmacokinetic properties, everything was described in this. So like this, you can check for any Indian medicinal plants, popular Indian medicinal plants. Okay, so this is something about how to use just checking to the plant or even checking to the compound. You will be getting the properties related to that. Other than that, we can also come to know the plant linked with the diseases information in this impact. Okay, so our company is also a part of is doing a part of services related to this. We can able to provide you a pure compounds. Okay, for more details, visit our website. Okay, so of course, because I'm explaining about this, one should have a knowledge of this phyto nanostruct technologies. The products what we have, I just want 30 seconds to complete. So our major products are root powders from these seven plants. Okay, and we can also see we are growing the herbs in the pots, as well as we are growing in the soil. So now we are trying to uh, increase even the plants, and we are going to increase the products in future. And to know about the major products whatever we are providing, other than the root powder, plant-based materials, we are also providing the secondary metabolites of above 50, above 80, above 95% purity. And these are the compounds at present we are marketing. Slowly we are going to increase up to 100 secondary metabolite products. And even from Ramachandra, three scholars are procuring the compounds from us. And I believe even from the Bharatidasan University, one more professor is procuring compounds from us. So if anyone want to procure the thing, okay, so you are welcome to contact us. Okay, so the website is pndfntech. Okay, I've completed my session. Any doubts you can ask me, and if anything is unexplored, we'll be continuing this in the subsequent sessions. Okay. 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 Any doubts you can ask me at present. So if no doubts, thank you very much for your interactions and patience. So I suggest you to practice and you try to apply in the research, produce in the form of meaningful outcomes, in the form of conference or journal publications. So you can also link with me through consultancy services or any kind of internship courses which I conduct in future, which can enhance your knowledge with your outcomes. Thank you very much. Have a great day.